The peptide hormones could have hundreds of amino acids, so they're bigger. The amine hormones are small molecules like the steroids. So B is a true statement. Is C a true statement? No, because we know of at least one steroid, cortisol, that is required. Is D a true statement? It is not, because remember, enzymes are proteins, and steroids require enzymes for their production. So to make the steroids, we need proteins, enzymes. So A and B are true, and C and D are false. I will post on Moodle this version of the PowerPoint with the answers. I will also post on Moodle exam 2 and my answer key to exam 2. That will be on Moodle. So that will be review material when you're getting ready for the final exam. Receptors. Many of the hormone receptors are GPCRs. We've covered GPCRs a lot. You should be able to tell me the story that is partially illustrated by this slide. Receptor, binding, conformational change of the G protein, GDP dissociation, GTP association to alpha, alpha separating from the receptor and from beta gamma, alpha activating, in this case, adenylyl cyclase. This is the classic GS story. GS activating, converting, hydrolyzing ATP to cyclic AMP diffusing, binding to the regulatory subunits of protein kinase A, releasing active catalytic subunits of pKa, which has multiple effects. Okay, so this story and GI, where all the G protein story was the same, that is to say, binding, conformational change, GDP dis, GTP binding, but the GI inhibits adenylyl cyclase, and so it reduces the concentration of cyclic AMP because of endogenous phosphodiesterases that are cutting cyclic AMP. So stimulating cyclic AMP, inhibiting the production of cyclic AMP. GS and GI. And those effects, either stimulating or inhibiting pKa, has multiple downstream effects. Those stories should be very familiar. Multiple hormones interact with the GS story, that is to say, their receptor. Now remember, their receptor is very specific. The TSH receptor is very specific for TSH and doesn't bind PTH at all. The receptors are specific, but all of the receptors for these hormones couple to GS. All of these receptors are very specific for their hormone, but they all couple to GI. I do not want you to memorize this list. If I want you to tell me the GS story, I will tell you that we're talking about a GS story. In the problem set, I don't tell you that because it's open book, and you can look up on this slide. So the problem set question 5 asks you about the 5-HT2 receptor. So, well, there's 5-HT1. Where's 5-HT2? 5-HT2 is here. It's a different story. Which story is this one? This is the GQ story, right? This one has more details. GTP binding, GDP dissociation, GTP binding, after conformational changes. Phospholipase C, you've heard this story. This slide has a lot of the detail of the GQ story. IP3, calcium signaling, calcium binding, activating either or both protein kinase C or other calcium binding proteins like calmodulin that activates a kinase. Multiple effects. Again, multiple hormones, all interacting specifically with their receptor, but their receptors are specific, but their receptors all couple to GQ. Do not memorize this list, but be prepared to use the GQ story. Meg?
When you ask us about one of these, will you tell us if it's protein kinase C or calmodulin that it goes to, or is it just like, should we just say it could go to either or?
If I tell you that it does not couple to, I will either say, tell me both final effectors, or I will accept either one as full credit. I will give you a hint as to whether I'm looking for both of these stories or just one. Questions about the GPCRs? You can see we're covering a lot of our hormone receptors, but if you understand the GS story, you understand the cellular effects of a whole bunch of hormones. If you understand GI, you understand the molecular mechanisms for a whole bunch more hormones. Interestingly, some receptors for norepinephrine and epinephrine are GI, and some other receptors for epinephrine and norepinephrine are GS. So the same hormones can have very different effects depending on what receptor is expressed in the target cell. Beta versus alpha adrenal receptors catalyze very different effects. So if I were to say, tell me the story of dopamine, what does dopamine do? I would have to say, tell me both details about GS and GI. I do not expect you to memorize that dopamine couples to both GS-coupled receptors and GI-coupled receptors. Okay? But question 4 on the problem set asks you to outline the story downstream from D1. So, there's the story. You can look it up and tell me the story. Why do I ask you to tell me these stories one more time? Repetition assists in formation of long-term memory. That's why. Okay, so GS, GI, and GQ. Interestingly, norepinephrine and epinephrine interact with GQ, GI, and GS coupled receptors depending on what receptor is expressed in the target cell. We talk about that more in AMP2. Okay, we're done with GPCRs. No, we're for the next half hour. You are not done with GPCRs. GPCRs are in your future just like they have been in your past. Now let's talk about a different type of receptor, the tyrosine kinase receptors. Receptor tyrosine kinase, or RTKs, a whole nother class of receptors. There are 3 hormones that couple to receptor tyrosine kinases. I don't think it's too much to ask that you memorize these 3, right? if you memorize those three, then I could ask you to tell me the story. So insulin, growth hormone, and prolactin, and we'll talk about their effects more in a few minutes, well, on Wednesday, all interact with receptor tyrosine kinases. I think you learned about this story in cell-phys. But let me review it. Binding, receptor subunit dimerization, forming a receptor hormone complex. Phosphorylation on tyrosines of the receptor subunits. So binding, dimerization, phosphorylation of the receptor subunits. That's what's shown here, phosphorylation of the receptor subunits. And then a whole cascade of effects. So this slide is a little bit ambiguous. So let's see, what are the effects that happen after, you know about this from cell-phys. What are the effects that happen after phosphorylation? We've got scaffolding of adapter proteins. And we have a kinase cascade. You've learned about the MAP kinase cascade from cell-phys. Somebody who took cell-phys this year, which I didn't teach, is this familiar? I just want to make sure. Did you cover the MAP kinase cascade in the fall of 2020? Okay, yeah. So you've heard this story. Dimerization, cross-phosphorylation, adapter protein binding. Did you hear about RAS? R-A-S. Okay, RAS binding, RAS activation of a kind of MAP kinase, and MAP kinase having multiple effects. That's what's missing from this slide. So I hope you have annotated your handout to indicate that this right in here, this cascade of effects requires adapter proteins, RAS, and a kinase cascade, MAP kinase. And then multiple effects. That's the receptor tyrosine kinase story. Building on cell-phys, using it for these three hormones. Questions? Okay, let's talk about the receptors that are not on the surface of the cell. The receptors are inside the cytoplasm of the cell. They're called cytoplasmic receptors. That's helpful. So all of the receptors that we've talked about so far, the GPCRs and the receptor tyrosine kinases, are located on the surface membrane, and they bind hormones from the extracellular solution, binding and then effects inside the cell. Now we're going to talk about receptors that are cytoplasmic proteins. So how does a hormone get, that's what the H is here, how does the hormone get from the blood to its receptor? What word begins with D is embedded in these two arrows? Diffusion. Simple diffusion of the hormone into the cytoplasm. Now, many of the hormones are carried by proteins in the blood, but not all of them. Some of them are just in the blood. But in any case, there is hormone in the blood, diffusion of the hormone through the membrane. So what does that mean? The hormone has to be at least partially hydrophobic in order to do this diffusion. So now we can tell we're talking about the steroid hormones, and it turns out the thyroid hormones also do this. So if you remember the names of the steroid hormones, which you do need to remember, you know 5/6 of the types of hormones that interact with this type of receptor. All of the steroid hormones, the sex hormones, cortisol and aldosterone, all of the steroid hormones, and the thyroid hormones follow this story. So I think I could ask you to know that. So basically, if you know the steroid hormones, and you know the thyroid hormones, follow this story, and you know that these three hormones follow this story, then everything else is a GPCR. Okay, sorry. So let's finish this story. Receptor binds the hormone in the cytoplasm because of diffusion. There is translocation of the receptor hormone complex, usually to the nucleus, because in most cases the receptor to these types of hormones are transcription factors. Changes the expression of mRNA, changes the synthesis of some protein. That is the response. So in this case, protein synthesis is the downstream effect due to a transcription factor changing, either up or down regulating transcription. In none of the other stories is protein synthesis such an integral part of the story. It might be. Protein synthesis might be required here. Protein synthesis might be required here. But protein synthesis is required here. Okay, now I want to introduce a general concept. So we're going to talk about a general concept, and we're going to be using this general concept over and over and over again as we get specific. A general concept is that we have positive feed forward. So one hormone stimulates another cell to produce a hormone, let's say, and another cell produces a hormone that has effects. So something from the brain might affect the pituitary. The pituitary secretes a hormone that affects something else, let's just say anything else. And there is feedback, positive feed forward, stimulation, production of something that changes the cell production, changes the cell behavior, phenotype, and negative feedback. So positive feed forward, stimulation, and negative feedback is going to be the common thread that we're going to see in order to maintain homeostasis. So hormones having positive effects and then negative feedback loops to try to shut down the hormone in order to maintain a balance. We've got a stimulation and an inhibition. The combination of antagonistic controls, just like in muscles. Stimulation of the biceps causes flexion. Stimulation of the triceps causes extension. If I stimulate them both to a certain extent, I get something in between. If I need to hold my arms like this, I have homeostasis with some contraction of my biceps and some contraction of my triceps in order to maintain this position. Homeostasis of this position. Homeostasis of hormones, positive and negative effects to maintain a balancing homeostasis. we're going to see that over and over and over again. So let's take a 5-minute break here while I transition to the next PowerPoint, because now we're going to get into some of the specifics. And it will be important because we're going to be talking about the pituitary, which, as I said, is an important master controller. So let's take a 5-minute break.